Welcome everyone once again to Zahid's data mining channel. My name is Zahid Islam, and today I will be talking about a genetic algorithm based clustering technique called GMC. This genetic algorithm based clustering technique is just presented and published in a IEEE CEC conference recently in Vancouver. Here you can see the full reference of the paper. The name of the paper is Novel Crossover and Mutation Operation in a Genetic Algorithm for Clustering, and it was presented in IEEE CEC in Vancouver only last week. So the paper may not be online yet, but it will be online, and you can find the paper online by simply Google searching. In case if you are having any difficulties in any problem downloading the paper, please do not hesitate to let me know. Email me and I will send you an author's copy to you. Okay. Now, this paper is on clustering. What is clustering? Clustering is actually grouping similar records in a group in a cluster, clustering similar records in one group and dissimilar records in different groups. So, for example, if this is a data set with few records and two attributes, as you can see on the screen, then these records can be actually plotted on a two-dimensional plane, because this is a two-dimensional data set. There are only two columns or two attributes in this data set. So once you have plotted them on a two-dimensional plane, you can easily see that there are three different clusters or three different groups in a way in this data set, and this is what clustering does. There are many existing clustering techniques. Some of them are known as hard clustering and some of them are known as fuzzy clustering, but these existing clustering techniques often will require user input from you. For example, k-means, a very well-known and commonly used clustering technique, will request you to provide number of clusters to it. Whenever you ask k-means to do clustering for you, it will say, okay, you tell me how many clusters are there. And often that is a very problematic thing for a user. How do I know how many clusters are there in a data set, right? In advance, I am asking you to do it for me. How do I know it? And other clustering techniques will require some other parameters. For example, densclust will possibly require the radius of the clusters, density of the clusters, etc. In order to handle that, people often use genetic algorithm based clustering techniques. They are generally parameterless. They don't need any user input, and they also try to avoid getting stuck at local optima, which is a problem for k-means as well. Genetic algorithms are not guaranteed to get global optima, but they are likely to get better local optima, if not the global one. And the main goals for clustering is knowledge discovery from a data set. Of course, if you can cluster the records, then you can learn about it more, and then you can learn some insightful knowledge about a data set. Here is an example of knowledge discovery that I'm presenting it now. It was done on a brain data set. The details or the link of the brain data set can be found here, and they collected 22 epileptic patients record at Children's Hospital Boston, and they implanted the EEG electrodes using International 1020 system. There were 23 channels or electrodes altogether. This is how they implant. This is the convention, and then each of these channel will actually observe or record EEG signal, which looks like this, and the signal can be a seizure signal or a non-seizure signal. If it is a non-seizure signal, then generally this is how the signal looks like. So in this example, we have just used 1 hour data of one particular patient. She is a young girl of 11 years age, and she experienced 40 seconds of seizure during this 1 hour. During these 3,600 seconds, there were a 40-second time window when she experienced seizure. So we converted these seizure signals into records where we use 10-second epoch, which means that every 10-second signal was converted into one record having three attributes: max, mean, and standard deviation. Say, for example, this is a 10-second signal. So this signal is converted into a record where we find the max value of the signal, mean value of the signal, and we calculate the standard deviation, and this is how we create one record. So now, because there are 40 seconds of seizure period, the window, there are 5 records affected during this 40 seconds, and these 5 records are now labeled as Caesar record. This labeling is done only for cluster validation purpose and not used in clustering purpose. For the clustering purpose, we are using only these 3 attributes, right? So if there are 23 channels and each channel has 5 Caesar records, so that means there are 115 Caesar records altogether, and Caesar signal will look like this, right? So the signal will be very erratic compared to a non-Caesar signal. Now we used our technique called CS cluster, and we clustered the records. We plot the records in a 3-dimensional plane and we clustered them using CS cluster, and this is what we get. We get 2 different clusters, and one cluster has 8,240 records. These are mostly non-Caesar records, you can see them, and the green dots or circles are cluster 2 that has Caesar time records. So cluster 2 has 40 records and all 40 of them are Caesar signals. Now one thing we miss here is that we found that there are 115 Caesar records, but how come this cluster is getting only 40? What we then explore from these clustering results, we investigated the data set a little bit more, and we found that although there is a 40-second of Caesar window, not all electrodes were actually experiencing Caesar type signal during that 40 seconds. For example, you can see that channel 7 during Caesar time, this is their signal, and this signal is more similar to a non-Caesar time signal compared to a Caesar time signal. So what happened actually, we investigated it further and we found that there were 9 out of 23 channels were only experiencing Caesar type erratic abrupt kind of signal during the Caesar time, and other channels were not. 14 other channels were not actually experiencing Caesar type signal, and more importantly, all these 9 channels are located in the frontal lobe of the brain, which tells us that this Caesar was a localized Caesar as we understand it at the moment. So that means our clustering technique could actually extract or group the right Caesar signals or right record as a Caesar record, and this was done very effectively. So that's how it discovers knowledge, and we then actually went further in exploring or extracting knowledge using our clustering result. What we did is we labeled the records into 2 different labels, Caesar and non-Caesar. Now this time depending on our clustering result, not just whether it falls within the time window or not. And then based on that we built a decision forest, a number of decision trees. Here in this case we built 4 trees using our decision forest algorithm called CIS4, and we found that these trees are very accurate in terms of classification. 12 out of 13 libs actually have 100% classification accuracy. Now I will tell you a quick example. If you have a student data set, for example, you have many information about each student, such as like how many hours he studies, how many lectures he attends, how many tutorials he attends, etc. And then at the end you have a labeling, class label for each of the student, either good student or bad student. And if you assign the labels randomly, okay, you pick a student without knowing what is his information, good, he studies a lot or not, etc. You just label him randomly, either good or bad, and then you label all the students in the same way. Then you try to build a classifier from it. What will be the classifier characteristics or properties or the performance in this case? It will be a disaster for the classifiers. For example, a decision tree will be very inaccurate because it will not find any relationship between the labels good or bad and the properties, how many hours he studies or etc., because the labels are done randomly. So in that case the classification will possibly exhibit 2 types of properties. One, it will be very inaccurate even in classification, forget about prediction, and it will also be possibly very deep because it will be exploring a lot to find the pattern. But in this particular case that you can see on the screen, we find that all the classifiers, all the trees are very accurate in terms of classification, and they are very shallow. So that tells us that the labeling was good, it was matching with the properties of the signal, and that in turn tells us that the clustering was good. So clustering was good, that's why the labeling was good. So what explains here is that a good clustering can actually extract very good knowledge from a data set. Now in this paper, in this example, we are proposing a new clustering technique called GMC, and now let me explain a little bit about a genetic algorithm based clustering technique. A genetic algorithm is uses chromosomes, right? What is a chromosome? Chromosome is a clustering solution. For example, in this example, you can see that these are the three clusters here, and each cluster can be identified by these red dots, which is the cluster center, and then each of these centers is actually called a gene, and then having these three genes together makes a chromosome. So we get a chromosome here. So the steps that genetic algorithms use is initial population. It can have a huge number of chromosomes. It starts with not just one clustering solution, a huge number of solution, and then it uses an operation called crossover, where a pair of chromosomes can participate in a crossover operation, and they generally exchange their genes. And mutation is like one of the chromosome or one of the gene in a chromosome can get changed a little bit. These are the main genetic algorithm operations. So here in this paper we propose a few new approaches. For example, for initial population, inspired by our previous techniques. Please read the details in the paper. All these things are very clearly explained in the paper, but just to briefly introduce, we introduce an initial population technique where the idea here is to actually start the crossover and mutation and the whole genetic operation process with a very good initial population. If you start with a very good set of initial population, instead of the way that traditional algorithms do is they randomly pick a random set of initial population. Instead of that, if you can start with a good set of initial population, the chances will be higher that you will get a good end result, something like that. We also use a probabilistic selection operation where among two generations probabilistically we pick the best chromosomes, good quality chromosomes for the next generation, something like that. And we also use a new crossover operation where we divide the chromosomes into two groups, good chromosome and bad chromosome, and that is consistent with the approach that traditional algorithms use, genetic algorithms use. For example, the Rowlett wheel approach for crossover also encourages to find out good chromosomes so that good chromosomes can cross over together. So we actually go one step forward in that direction. We find out a set of good chromosomes and bad chromosomes. And how do we find out the good chromosomes? We actually find out the influence area of the best chromosome, and any chromosome falling within that influence area is grouped as a good chromosome. And then we encourage huge crossover among the good chromosomes. So huge crossover means, like, instead of picking one best chromosome and any one good chromosome, roughly probabilistically, we say, okay, let us have crossover among all chromosomes, all chromosome pairs of this set of good chromosomes. And this is how we encourage more crossover within the good chromosome set. And then finally we probabilistically again pick the best three chromosomes. And also we do the same thing for bad chromosome, but a little bit less extensive crossover. Now we extend the similar kind of approach for mutation operation as well. We again find a good group of chromosomes and a bad, relatively not so good group of chromosomes, and then we do some sort of division and absorption where we divide any sparse cluster into two different clusters by using k-means and using k equals 2, and absorption, when we find two clusters very close to each other, we generally combine them. That's what we do. And for good group, we don't use a random sort of mutation because the group is already very good, so we don't want to disturb it. So please feel free to read the details in the paper. Due to time limitations, I cannot go through the details, but but we have actually experimented our technique on 10 existing techniques, and this is the result. The results are also presented in the paper in details, and we can show that our technique actually performs very well, and it's better than the 5 existing techniques that we used in this paper, and we came up with very good clustering results. And we also compared all these individual components in experiments, so that to find out the usefulness of these components. Okay, that's more or less is. Thanks for your time. Thanks for